Hey guys, it's Shreyas here and welcome to Simple Biology. In this video we're going to be diving into the nucleus and learning about all the different parts of the nucleus and really getting a better understanding of what the nucleus actually is. So let's just get right into it. So my strategy for this video is that I'm going to be focusing on the very outside of the nucleus and work my way inwards, okay? So let's go ahead and just visualize that. So imagine we have a cell here. So we have the cell membrane, okay? This is the cell membrane. And then we have the nucleus on the inside, okay? Now, the nuclear envelope, which is right here, this is the nuclear envelope. Also, sometimes it's referred to as the nuclear membrane in textbooks. It means the same thing. But the nuclear envelope or the nuclear membrane is basically what this membrane which contains everything which is in the nucleus. We know that for a cell, that the cell membrane kind of holds everything, all the organelles of a cell together in one region, kind of packs everything together. In the same sense, the nuclear envelope holds all the contents of the nucleus together. It keeps the DNA and other materials close. Okay. Now what's interesting about a nuclear envelope is that it's a double layered membrane. What does that mean? It actually consists of two lipid bilayers. If you don't have any idea what a lipid bilayer is, you might want to go and check out our video on membrane structure. Okay, we have a video on membrane structure somewhere throughout this cells unit. Go check that out. Okay, so as you can notice in this diagram here, we have again we have a kind of a snippet of a cell. So here is the cytosol, and then here is the nuclear envelope, right? So what if you look closely here, there's actually two lipid bilayers. There's one right here, so this is a lipid bilayer. And then there's another lipid bilayer on this side. And in between these two lipid bilayers is that there's space. Okay? Now, the nuclear envelope also has pores. It has scattered pores. If you look in this diagram here, you'll notice that there's these holes right here. Now, it might be kind of hard to see, but these pores are actually ringed with protein. So what I'm going to do here is I'm going to go ahead and say we take like a cross section of this area and we actually try to visualize it. Okay, so if we took a cross section of this area, what we notice is that there would be again the two lipid bilayers. So this is one lipid bilayer, and then this is another lipid bilayer. And then what we'd also notice is that we would see this ringed protein. Okay, it's kind of bad here. So this ringed protein, you have to kind of imagine it in a 3D view. This is kind of like a this ringed protein here. So it's actually 3D. You have to kind of imagine it like in a cylindrical shape, you know. So this ringed protein is actually let me write this down. This is a protein, very important to know. This ringed protein is going to determine what goes into the nucleus and what goes out of the nucleus. Okay, so if something can pass through and the proteins are okay with it, then obviously the protein is just going to open up and the pore is going to open up, allowing for that material to pass in or pass out. But if the protein is not okay with allowing that material to pass in or pass out, the protein is going to close up and the pore is going to close up, and that material is not going to be able to pass against the nuclear envelope. Okay, and that's so critical towards a nucleus because remember that the nucleus is the brain of the cell. You know, I mean, that's kind of a immature way of putting it, but it really is just like the part of the cell which has all the directions for the cell. So if the nucleus, you know, gets a pathogen inside of it, it's bad because then the whole cell will die, because without your brain, you know, you will die. In the same sense, without the nucleus, the cell will die. Okay, so it's very important that the cell is able to the nucleus, is able to regulate what goes in, what goes in, and what goes out. And these proteins do exactly that. Now, before I move on to the next part of the nucleus, I want to mention how the nuclear envelope is continuous. Let me write this term down. Continuous with the ER, with the endoplasmic reticulum. So if you look at this diagram here, this is a kind of a snippet of a cell too. So here we have the ER. the endoposic reticulum, and you can see we have the smooth ER with the ribosomes. I mean, sorry, the smooth ER without the ribosomes, and the rough ER with the ribosomes. And then here we have the nuclear membrane. Okay. Now the interesting thing is that there's nothing separating the nuclear membrane from the ER. Okay, so if you look here, I'm going to just take away the highlighting. Remember that the blue shaded region is the nuclear envelope, and then the yellow shaded region is the ER. So I'm just going to take this away. So if you look here, that these regions are actually connected at these places. So that's what it means to say that the nuclear envelope is continuous with the ER. There's nothing which is separating it together. These regions are actually connected. The nuclear envelope is connected with the endoposic reticulum. Okay, very important that you know that. Now let's go a little bit more. So again, if you look at our generic nucleus diagram, we have the nuclear envelope. But then as we get a little bit deeper into the nucleus, we actually noticed this nuclear laminas. Okay, it's kind of just kind of bordering the nuclear envelope, kind of like this. Okay, let's try to get it in here. Okay, just bordering the nuclear envelope. You can also look at our diagram here. So this right here, this purple line right here would be the nuclear envelope, and then this right here, all this mess here would be the nuclear lamina. So what is a nuclear lamina? The nuclear lamina is actually a network of protein strands. Okay, as you can see here, there's a bunch of, it's just kind of weaved in together here, and that's essentially just proteins are just weaved in together. And the function of the nuclear lamina is that it maintains the nucleus's shape. Okay, just to give you a simile here so you have a better understanding of this, I want you to get up, and I want you to go punch a wall. Okay, don't actually do that. Okay, but if you've ever looked at a wall and you actually went and punched it, you notice that there's two parts to a wall. There is the dry sheath. Okay, the dry sheath, which is basically kind of like the white part of the wall, which you see. You see the paint, and then there's like this very thin layer, which kind of supports the wall, giving it its that pretty white color. But underneath the dry sheath, you see these long blocks of wood, and the wood is what gives the wall its actual strength. So if something if wind or if something comes and it hits it, the wall is not just going to completely fall down. Okay, so in the same sense, when we talk about a nucleus, the dry sheath of the nucleus is kind of like the nuclear membrane or the nuclear envelope, same thing, and then the wood is the nuclear lamina. It's the thing that's giving it that support. Okay, I hope that simile helps you understand this. Okay, now there's actually this interesting information which says that some new research, apparently some crazy research, which says which suggests that the nuclear lamina extends through the nucleus. So what does that mean? So instead of just having the nuclear lamina kind of on the outside like I drew here, some evidence suggests that the nuclear lamina actually goes everywhere through the nucleus. Okay, it's not just bordering the nuclear envelope, it's actually everywhere. Okay, and what would that mean? That would mean that maybe the nuclear lamina, actually helps organize genetic material. So as we know from pre-AP biology, the nucleus consists of genetic material, and the genetic material is actually found, kind of in this inner region of the nucleus, which we'll get into in just a second. But if the nuclear lamina goes completely, this network of proteins goes completely throughout the nucleus, instead of just bordering it, maybe the nuclear lamina also helps organize this genetic material in the center of the nucleus. Okay, now this is not completely verified. Scientists are still doing research on that. Maybe you can go, if you're in your own free time, go look it up on Google and see if this is actually true or not. But scientists know for sure that this is true. This is still in question. Okay. Now let's go a little bit further. So again, just for emphasis, we have our generic nucleus diagram. We have the nuclear envelope, then we have the nuclear lamina bordering right outside. And now if we dig a little bit deeper, we will find the genetic material, the chromosomes and the chromatin. Okay, so I'm just going to draw in chromatin first. So chromatin is basically just like this diffuse genetic material. Okay, so chromosomes and chromatin are made up of DNA. Now, if you have no idea what DNA is, that's really bad. I would highly suggest that you go watch our biomolecules unit. DNA is very important. You need to know what that is. So the chromosomes and chromatin are both made up of this biomolecule DNA, and they're also organized by proteins. Okay, whoops, I need a highlighter. Organized by proteins. Okay, now what's the difference between chromatin and chromosomes? Chromatin is diffuse. Okay, chromatin is diffuse. So here we're looking at chromatin. In this diagram here, we're looking at chromatin because the genetic material is kind of just diffuse. It's just spread all the way throughout the nucleus. There's not that much organization to it. So whenever the genetic material is in the chromatin form, the cell is not dividing. This is very important that you know, not dividing. Okay, and actually, if we looked at cells under a light microscope, and we were not able to see anything in the nucleus, we would know that the cell is not dividing because chromatin is so diffuse, you know, you're not going to be able to see anything under a light microscope. Okay, so a test question which might be asked to know that. However, whenever the cell is about to divide, so when the cell is dividing, this chromatin is going to be converted into chromosomes. Okay, and chromosomes are actually very condensed genetic material. So let me go ahead and draw what would that would look like. Okay, so we have the nuclear envelope, and then we have the nuclear lamina surrounding the kind of bordering the nuclear envelope, and then we're going to see this very distinct shapes. These right here are chromosomes, so we'd actually see these chromosomes, and under a light microscope, we would actually be able to see these chromosomes because they are so dense. Since they're so dense, it's actually easy to visualize. So if you looked at a cell and you wanted to know if it was dividing or not, if you see chromosomes, you see these interesting shapes, these X's, you know, you know that cell is dividing. But if you don't see anything, then you know that the genetic material is in the form of chromatin, and the cell is not dividing. That's something important that you should know. Okay. Now, before we move on, there's actually two parts of a chromosome which you need to know. This end here, these ends of a chromosome are telomeres. Oops, are telomeres, and the very center here is called a centromere. Okay. Now we're going to be diving way more into depth on chromosomes and chromatin in later videos. Actually, in our genetics unit, we will be focusing so much on the chromosomes because the chromosomes are the basic unit of genetics. But for now, this is all you need to know about chromosomes and chromatin. Okay. Now, the final part, if you really dive in, again, I'm going to do this one more time just for emphasis. So again, we have the nuclear envelope, we have the nuclear lamina. Okay, every time I draw this, I feel like it gets worse and worse. Okay, and then we have the chromatin. Okay, like this, the chromatin. Okay, and then finally, if we dig even deeper, we will be able to notice this final part of the nucleus called the nucleolus, which is going to be like this. Okay, well, before we do this, I just want to take a step back and make sure you understand this, okay? So we have one nucleus right here, and then we have one nucleus right here. So I want you to find out which one is dividing and which one is not. Just take a second and do that. Okay, so this nucleus is part of a cell which is not dividing, and this nucleus right here is part of a cell which is dividing. How do we know that? Because here we have the chromosomes, so since we have chromosomes, the cell is in division, and since here we have chromatin, the cell is not dividing, okay? So make sure you understand that, very important, all right? So anyways, the nucleolus is this region right here. There's a nucleolus. Here I drew another nucleolus, and what you need to know about the nucleolus is two things. First, it is not membrane bound, okay? So just like how we talked about how the cell membrane is the cell is bound by cell membrane and the nucleus is bound by the nuclear envelope, the nucleolus is not membrane bound. It's kind of just like floating around like this, and it's the region of the nucleus where ribosomes are manufactured. Probably the most important part. You can call the nucleolus is the ribosome factory for a cell. It's where the ribosomes are made. Okay, that's all you have to know about the nucleus for now. Hope this video helps. I would really appreciate feedback on what I could have done better. But that's it for now. The nucleus is as simple as that.